So the current vaccines being put out were put out so quickly, like historically fast, right? Is that safe? Is this something? Should we trust a vaccine that's been sort of fast tracked so quickly that's beyond normal historical record?
Fast vaccines come very quickly, but they're based on technologies that are very novel. So what these technologies do, they cut down development time quite significantly. Normally what you want to do a vaccine, you would take the virus, biological material in which the virus is contained, you put it to a process that could include killing the virus or attenuating it through different passages, through different stages. And then you have that growing into a medium, which normally is eggs or egg proteins or some kind of live biological material. And then when you have enough of it, you have to extract it and purify it. And then you need to take that and then test it to make sure that is purified and clean and safe, and then you move it to the next stage and to the next stage and to the next stage. And those stages include in vitro, looking at reactions into a petri dish, to animal experiments when they look at effects into a living organism. It could be rats, mice, dogs, chimpanzees, monkeys, all kind of different animal models. And then finally, they come into the clinic where they're tested first in healthy volunteers and then in different groups of patients. So the build-up piece, the one that goes from identifying a potential vaccine to getting into your in vivo experiments, so even the animal toxicity, takes years because it's really, really tough to purify a virus and have it stable enough that you can process it in a reliable way in large quantities so that it can be available as a vaccine. And then you have to do all the toxicology piece to make sure that that is not infective, right? So that you don't actually give somebody the disease by giving them a death of attenuated virus. Because this process was very inefficient, new technologies were discovered.
Invented.
And one of those new technologies was, well, why do I have to take the whole virus, right? Why don't I just take the protein from the virus that causes the immune reaction and against which the antibodies, when somebody has a natural infection, are developed, so that I can skip out, all the virus, dealing with the whole virus altogether. And this is protein vaccines. So these are protein vaccines, and this were the latest generation of vaccines before this last bout. And those vaccines were still kind of laborious to do because you had to genetically modify normally bacteria to produce the protein. Then you had to scale up the process so that you can have reliably, tons and tons of bacteria creating the same protein without contamination. And then you had to purify the protein and then put it into a vehicle and then, still running through the in vitro and in vivo testing. Now, the latest generation is genetic vaccines. So those vaccines work like the AstraZeneca vaccine, for instance, or the Janssen vaccine. They work in a different way. They take a virus, which is normally a harmless virus. In this case, both use, I think, adenovirus, which is one of the viruses that causes the common cold. So they have very low, they're very high infectivity, but very low pathogenicity. So they don't cause a lot of a disease, right, in the majority of people. So those viruses are further modified to receive a piece of DNA which expresses only the protein against which you want to have a direct response. So in a way, those are kind of cutting out the middleman, right? They're cutting out us creating protein in a vat through bacteria in a vat. You just put the genetic code directly into a virus, and then you let the virus go into the cells and do its job by, by creating the protein. The protein will be secreted normally as any cell would do once it's infected by a virus, and create an immune response.
And that's what we have right now? That's the mRNA vaccine that's being
that
put
is
out?
the AstraZeneca or the Janssen vaccine, for instance. The Pfizer and Moderna vaccines are a step above. So they just say, you know what, I don't even want to go to the trouble of putting this thing into a living organism, even if it's attenuated. I'm just going to create the blueprint, the already available transcribed genetic material, so that it doesn't even have to go through the elaborations of DNA. It just has to go into this organelle that is called ribosome, which is in every single cell in the human body, and because it's a blueprint, it's gonna just be translated directly into the protein. And so we cut the middleman, and we cut the middleman. We basically cut all the steps. We just kind of synthesize a very small piece of messenger RNA. M stands for messenger. And the messenger RNA is essentially the part of genetic code that is able to come out of the nucleus of a cell and be translated into a protein.
So that's why we're capable of doing it so quickly this year, is because the technology has been invented to do it quickly.
Right. So the technology allows you to cut out all of the purification piece, all of the extraction, all of the protein synthesis. You just have your own cells to do the job for you.
Yeah.
If you want, it's a more natural way of actually doing it. This is what you would do normally, right? You get infected by a virus, your immune system recognizes the proteins from this virus, and they mount an immune response. In this case, you just have your cells producing the proteins. And with mRNA, which is different from DNA, you actually only have a limited amount of the genetic material that you give. And when that is gone, that's gone. And that's why you need two shots.
Oh, interesting.
Right? Because one, you might not have enough material to mount a significant response.
So in a way, what you're saying, what I think I'm hearing is that the vaccine developers essentially just sort of did a shortcut of the process that the body would do.
Right.
Naturally.
Right.
Interesting.
And again, with an mRNA vaccine, you have no chance of genetic modification. You have no chance of insertion of the genetic material. So every time you use, for instance, a virus, you have a small chance that some of the genetic material of the virus will be inserted into your own genetic material. Without having a virus, and without having any other genetic material there but messenger RNA, there is no chance of any insertion of mutagenesis or changes in the host DNA. It's literally a blueprint.
Wow.
To do only one thing, and one thing only.
Which is?
Which is to synthesize the spike protein from SARS-CoV-2.
Which does what in the body?
Well, which is what creates the immune response and prevents the virus from attacking the body.
And that's we've seen that happen in patients so far, where when the T cells are boosted, right?
There are two responses. So you have the antibody response, and then you have a cell response, and they're not the same. So with COVID, we're actually learning that T cell immunity might be just as important as B cell immunity. We normally measure immunity by antibodies. But for instance, with COVID, we see a lot of patients that recover from COVID not developing antibodies or having very short-lived antibodies. But yet they don't get reinfected, and that seems to be because they mount a T cell response.
And that in and of itself means other questions, right? About the long-term effect of this particular COVID-19, that we may be able to achieve herd immunity on it, or we might not.
That's an unknown. We don't know whether even the vaccines will provide long-term immunity. So far, we have 9 months data, and we're coming up to 1 year, and hopefully, we will have immunity lasting for longer. That is not novel, that is not new. For instance, with tetanus, you need to have a recall from a vaccine every 5 years, right? With other vaccines, you might, with the flu, for instance, because it changes so quickly and so dramatically, you have to do it every year. With COVID, we don't see that amount of mutation in the virus, but we don't know yet how it fully interacts with the immune system. So if anything, my concern with the virus is that since we don't fully understand the immunological effects of COVID infection, that could be related to long-term effects, could be related to autoimmune reactions and so on and so forth, we don't fully understand whether some of those effects could be also carried out by the vaccine.